So now let us see the structure of the male reproductive system. In human beings, the testes are extra-abdominal. Extra-abdominal means outside the abdominal cavity. There is a reason for this. When they are found outside the abdominal cavity in pouches of skin which are known as scrotal sacs, they are at a temperature which is 2 to 2.5 degrees lower than the body temperature. So, advantage of this extra abdominal sex organs or testes is because of this they are maintained at a temperature which is 2 to 2.5 degrees lower than the body temperature. Now, how does this lower temperature help? It helps in spermatogenesis. At body temperature, the cells which actually produce sperms, they get damaged. So, this slightly lower temperature is required for spermatogenesis. So, this lower temperature is required for spermatogenesis, very important. So, where are these testes formed? They are formed in the abdominal cavity. When a male fetus is growing in the mother's womb, testis, the organ, is formed inside the same cavity, that is abdominal cavity. But in the 7th month of gestation period, that means the fetus is still in the mother's womb and it is in the gestation period 7th month. In 7th month of gestation period, these testes, they descend into scrotal sac. That means they are formed in the abdominal cavity, then they will move down into the scrotal sac. So, in 7th month of gestation period, these testes descend into scrotum or scrotal sac. This descent is controlled by a hormone called testosterone. So, descent is controlled or regulated by testosterone. So, 2-3 important things. Testes in human males are extra-abdominal. They are found outside the abdominal cavity in scrotal sacs. What is the advantage of this? Because of this, they are maintained at a temperature which is 2 to 2.5 degrees below the body temperature. Why this lower temperature is required? It is required for spermatogenesis. The cells which give rise to sperms, they get damaged at a higher temperature. So, if testes remain inside the abdominal cavity, then the spermatogenesis or sperm production will not take place. We will talk about those abnormalities and what those conditions are called later on. These testes, though they are found outside the abdominal cavity, when the fetus is growing, they are present or formed in the abdominal cavity. In the 7th month of gestation period, these testes, they descend into the scrotal sacs under the influence of testosterone. So, if by any chance testes fail to descend, spermatogenesis will not take place because of higher temperature. So, this is important in terms of the testis, its formation and then its descent. There is an important reason why they should be outside the abdominal cavity. Now, these testes, they are small bean shaped or little oval structures, approximately 4 centimeters by 2.5 centimeters by 2 centimeters, and they are present in scrotal sac. There are 2 scrotal sacs, one on each side between the legs. This scrotal sac is made up of loose skin, and there is a scar which is visible between these. This is what we are showing because we want to draw this, but if we actually show the scrotal sac, there are 2 and there is a connection between the 2. This is known as septum scroti. So, there is a scar-like connection. Because we want to see the structure in detail, we have separated these 2, but they are connected like this. The scrotal sacs are lined with smooth muscles. These muscles are known as Dartos muscles. The name of the muscles which are found in the wall of scrotal sac is Dartos, and Dartos muscles help in thermoregulation. This temperature regulation is called thermoregulation. So, let us write this word here, thermoregulation. So, the muscles, they help in this thermoregulation. Inside these scrotal sacs are present 1 testis each. So, 1 scrotal sac would have 1. So, let us draw this testis and then we will see the enlarged structure also. We can draw only in one, same thing because we are bilaterally symmetrical, everything would be same on the other side also. So, if we enlarge the testis, each testis has a fibrous capsule or cover which is known as tunica albugina. Tunica albugina. This tunica albugina on the inner side is thrown into folds, and because of these folds, the inner side or inner compartment of the testis is divided into many smaller compartments or locules. Each testis has about 200 locules, number is slightly variable. So, these are the locules. Outside this tunica albugina, there is a layer of flat celled membrane which is known as tunica vaginalis. It is a peritoneal layer. Is called tunica vaginalis. Inside each locule, there are some coiled structures, and the number of these coiled structures is variable from 1 to 3. So, in each there can be 1 to 3 coiled tubes. These tubes are known as seminiferous tubules. So, these are coiled tubes, and just to understand the structure we are showing only one in each so that we understand the structure, but there can be 1 to 3. Let us make 2 here. So the coiled part is here. If we take a section of this, we will see those cells where the spermatogenesis will take place. So that we will take up little later. Now, these seminiferous tubules in the locule region, they are coiled. When they come in the middle part, they become little straight. So when they reach here in the middle region, they straighten up, and these branches, they join to form a grid-like pattern. Grid is basically a geometric pattern. So we can see here these are highly coiled, and here they become more or less straight, but they are connected to form a grid-like pattern. This grid-like pattern, or these straightened tubes, make, or together they are known as retetestis. Retetestis. All these branches, they open up by very thin, again straight tubes into the epididymis. So the structure which is going to come here is epididymis. Now we are drawing it in this manner, but this epididymis is a highly coiled structure. So we will talk about this epididymis also in a minute. So what is happening so far is, in the testis, the fibrous capsule is known as tunica albuginea. These albuginea membrane folds are seen inwards, which divide the testis into many locules. Each locule has 1 to 3 coiled seminiferous tubules. Let us label this seminiferous tubules. These tubules are coiled in the locular region. When they come towards the center or the inner side, they become straight, and all these ducts, they join to form a grid-like pattern which is known as retetestis. And from the retetestis, some fine branches, they pour or put that secretion, whatever is going to be produced here, into another structure which is called epididymis. So this is epididymis. We are drawing it in the form of a structure which is not coiled, but actually it is a highly coiled structure. It is about 6 meters long. If you straighten it, it is a very long tube. It has 3 parts. The upper part is known as the head or corpus major. There is one more term, we will write that. The middle part is known as the body or corpus major. Okay, this lower is known as caput or corpus minor, and here it becomes a narrow tube which will join vas deferens. So we have shown a color change. This is vasa differentia. Sorry, this is called cauda, C-A-U-D-A. This is called caput, this is called corpus, and this is called cauda, the tail region. Okay, one more word is tail region. So, three parts. These ducts which open into the head region of epididymis are known as, these tubes, they are known as vasa efferentia. So now the sperms which are produced in the seminiferous tubules, they go through these ducts, come to retitestis, then with the help of vasa efferentia they are poured into the head region of the epididymis. Epididymis highly coiled, but it remains compressed near the testis, and there are three parts: head region, body region and tail region. The sperms are poured into the head region of epididymis. In epididymis, the sperms will be partially stored and they attain functional capabilities. So now let us show the epididymis here. We have seen the internal structure. So let us show this epididymis. The epididymis is on the posterolateral side. And a tube, it connects from the lower part and it goes up. When the testes, they descend into the scrotal sacs, that means they are coming from the abdominal cavity, how will they remain in their position? To keep them in their position, at the bottom of it, that means between the bottom of the testis and the wall of scrotum, there is a fibrous tissue which is known as gubernaculum. Gubernaculum is white fibrous connective tissue which attaches testis to the wall, a basic lower wall of the scrotal sac, so that these testes, they remain in their position. In non-mammals, there is a mesentery which is known as mesorchium, which helps holding the testes in the abdominal region. Okay. The cavity which is present here, the cavity that is between the scrotal sac wall and the testis, is known as vaginal coelom, and it is filled with connective tissue called hydrocele. So this cavity is filled with a connective tissue. Now let us talk about this structure again. The two scrotal sacs are connected. There is a connection here which is called a septum, septum scroti, and here they are connected with the abdominal cavity. So above this there is this abdominal cavity. So the connection or the passage between this hydrocele, or rather vaginal coelom, and the abdominal cavity is known as inguinal canal. So inguinal canal is the passage between abdominal cavity and vaginal coelom. So when the testes descend from the abdomen, they come into the scrotal sac through this narrow passage which is called inguinal canal. So now from epididymis, vas deferens, it ascends. That means it goes up and it goes into the abdominal cavity. Now let us draw bladder also here, the urinary bladder. So now this vas deferens or vasa differentia, it ascends, goes into the abdominal cavity almost up to the top of bladder and then turns downwards. So it goes up and then comes down. When it is bending, when it is coming down, at that place it shows a slight swelling which is known as ampulla. Ampulla of vasa differentia. At this ampulla region, there is a duct of another gland which opens here. It is a tubular gland which opens into vasa differentia. This gland is known as seminal vesicle. So from the testis, the sperms are poured into epididymis. Here they will attain functional maturity. From the epididymis, a duct is going to carry them. This duct is known as vasa differentia. The duct goes up. Now it is moving upward. So this duct ascends into the abdominal cavity, goes up to the top of the bladder and then bends downwards. At the bending, there is a slight swelling which is known as ampulla. Just at the base of ampulla, there is an opening of a gland called seminal vesicle. Now this vas deferens, it opens into this tube. The function of urinary bladder is temporary storage of urine. This duct is going to carry urine. So this duct is known as urethra or urinary duct. Now this vas deferens from one side has opened into this, and same thing will happen from the other side also. So two vas deferens are coming from their respective sides and opening into this tube. Now this later part of the tube will carry two things. It will carry the urine from the bladder and the sperms from the two testes which are being transported by the vasa differentia. So this later part of the tube is known as urinogenital duct. Urino because it is going to transport or transfer urine also, and genital because now it is going to carry the gamete, that is sperms also. This urinogenital duct passes through a muscular structure which is the external genitalia that is penis. The opening of penis is slit like and is known as glans penis. The skin which is present near the tip is known as prepuce or foreskin. It is called prepuce or foreskin. So the tip where the opening is known as glans penis. The tip is surrounded by loose fold of skin which is called prepuce or foreskin. One more gland, that is prostate gland. Prostate gland is present at the junction of the vas deferens with the urinary duct. So, and it is a donut shaped gland, that means there is an opening in the middle. So, if we just draw this, this is prostate gland. So, unless and until this prostate is removed, these connections are not going to be visible. But that is why we made the connections first and then the gland, so that we know exactly what the connections are. So, vas deferens from each side open into the urethra, and now urethra will be termed as urinogenital duct, because it is a common passage for the urine from the bladder as well as the sperms from the testis. At the base of penis, there are these small glands which are known as Cowper's glands, and these Cowper's glands, they secrete mucus. Cowper's glands are present at the base of the penis. So, the structure structure of male reproductive system, two testes present in the scrotal sacs, and we said that the scrotal sacs are not far from each other, they are together, there is a septum which separates them, but because we wanted to see the detailed structure, we made them separated. Each scrotal sac has a cavity which is known as vaginal coelom filled with a fluid known as hydrocele. The testes descend into the scrotal sac in the 7th month of gestation period under the influence of testosterone. Once they descend, there is a fibrous tissue which is formed at the base. It connects the testes to the wall so that they do not move from their place. There is one more structure which we will not be able to draw here. It is called spermatic cord, which is also a connection, and that also helps in thermoregulation. So, along with the smooth muscles of the scrotum, so smooth muscles and spermatic cord both help in thermoregulation. And gubernaculum is the structure which holds these testes or in their position. They do not let them move. What is inside the testes? Their wall is tunica albuginea, which is fibrous, thrown into the folds inside, so that there are compartments which are known as locules. Each locule has coiled tubes known as seminiferous tubules. Each locule has 1 to 3 tubules. In the locular region, these tubules are highly coiled. When they come towards the middle of the testis, they become straight and join each other in a grid pattern. This is known as rete testis. From rete testis, there are some 7 to 10 very fine ducts, vasa efferentia, which pour these sperms into head region of epididymis. Testes on the outer side are surrounded by a layer of single cells which is known as tunica vaginalis. Now, let us talk about what exactly is present in these seminiferous tubules. So, if we make a section, it is a cross section of we can say testes. If testes are cut, then these seminiferous tubules will get cut at many places, and what we will see is these cut seminiferous tubules. These seminiferous tubules are lined by an epithelium. This epithelium is the germinal epithelium. There are two types of cells which we find. One type are cuboidal cells and the second are long pyramidal cells. These cuboidal cells undergo meiosis to produce the sperms. That means these are the cells which will help in spermatogenesis. And these long pyramid shaped cells are known as Sertoli cells. So there are two types of cells here. Cuboidal, which undergo the process of spermatogenesis. So this is known as the germinal epithelium, and in this epithelium there are Sertoli cells. Sertoli cells play a very important role. The role, or let us write down what Sertoli cells do. They provide nourishment to the developing spermatozoa, or all those intermediate cells which will be formed during spermatogenesis. Nourishment is provided by the Sertoli cells, and that is why they are also known as nurse cells. They provide nourishment to the developing sperm. Second, they secrete proteins which are androgen binding proteins. Androgens are the male sex hormones, one of which is testosterone. So these Sertoli cells will produce some proteins, and these proteins will bind to testosterone, so that testosterone concentration can be maintained, which is required for spermatogenesis. Now, why these proteins are required? Testes only are producing these hormones. Now let us talk about the cells which produce testosterone, and then we will come back to this. Between these seminiferous tubule is connective tissue, and in between these seminiferous tubule there are cells. These cells are known as Leydig's cells or interstitial cells. Leydig's cells or interstitial cells. They produce testosterone. Now, the location where testosterone is produced is in the interstitial fluid outside the seminiferous tubules, and the location where sperms are produced is inside the seminiferous tubules. So, the hormone, that is testosterone, is required in here, but it is produced outside. So, these androgen binding proteins will help in binding with this testosterone. So, testosterone, which is produced by the Leydig cell, will move in, and these proteins will bind to it, so that the concentration of testosterone can be maintained very high. That concentration is required for spermatogenesis, which takes place inside the seminiferous tubules. So, if we take a cross section, this is what is visible to us. Many seminiferous tubules cut. In between them there are Leydig cells or interstitial cells. The germinal epithelium has two types of cells, the cuboidal ones which undergo spermatogenesis, and the pyramid-like ones which are called Sertoli cells or nurse cells, because they provide nourishment, and they also secrete androgen binding proteins. So, this is what is in this seminiferous tubules. Now, one more thing which we need to see is the cross section of the external genitalia. We did not label it, the structure is called penis. So, if we take a cross section of penis, then what is visible to us? Okay, so let us now talk about some more things which are there, and let me just erase a little part so that all these diagrams are as it is. So now keeping these diagrams as it is, let us see the cross section of the penis, the transverse section of penis. It is a highly vascular erectile tissue or organ. So if we cut it, there are 3 highly vascular erectile tissues which are seen. On the posterior side, there are 2. They are highly vascular, and when we take a cross section, we find only blood vessels cut. So all these circular things are the blood vessels which have been cut when we took the cross section of this structure. And then there is connective tissue. So it is highly vascular, and it becomes hard and erect because of this tremendous blood supply which goes here. These 2 are known as corpora cavernosa, and anteriorly there is one which is also highly vascular, but there is an opening which is seen inside this. There is a duct which is cut. This is actually the urinogenital duct. And outside this, again, because it is highly vascular, these blood vessels which are cut and the connective tissue. This tissue is known as corpus spongiosum. So out of all the erectile tissues, the urinogenital duct passes through corpus spongiosum. So this duct or this urinogenital duct which we have shown here actually passes through this, that is the corpus spongiosum, and all other place is occupied by connective tissue. So this is again connective tissue. So now let us wrap up the functioning of all the organs of male reproductive system. A pair of testes, these are the primary or main sex organs. They perform 2 functions. Sperm formation, we saw sperms are produced in the seminiferous tubules. So they help in sperm formation and hormone formation, the male sex hormones. These cells, which are Leydig cells, they produce male sex hormones, that is androgens. The most important one is testosterone. The sperms which are produced, they are passed into another structure which is called epididymis. Epididymis is a long 6-meter long tube, but it is highly coiled, 3 parts: head, body and tail. From the testes, with the help of fine tubes which are known as vasa efferentia, the sperms are poured into head region of the epididymis. In epididymis, the sperms are stored for a short period of time where they attain functional maturity. Then from epididymis, the sperms are taken by a tube which is known as vasa efferentia. This tube arises from the base part, that is the tail part of epididymis. Then it goes up into the abdominal cavity. In the abdominal cavity up to the level of the urinary bladder. Then it bends. The place where it bends, it swells a little bit. It is called ampulla region. And just at the base of ampulla, a gland opens into it with the help of a duct. This gland is a tubular gland. It is called seminal vesicle. Now, this vas deferentia, it opens into urethra. So, 2 vas deferens of 2 sides open into urethra. Up till now, that duct which was coming from the bladder was called urethra because it was carrying only urine. After the vasa deferentia open into it, now it will carry 2 things, urine as well as the gametes. So, it is now known as urinogenital duct. This urinogenital duct passes through the external genitalia, a muscular erectile organ called penis. The opening is slit-like, very tender, and it is known as glans penis. The tip also has loose fold of skin which is known as foreskin or prepuce. At the base of the penis, there is a pair of small peanut-like glands called Cowper's glands. One gland, which is a large gland, donut shaped or chestnut shaped gland, which is surrounding the junction of the two vas deferens and the urethra. Their secretions we will talk about a little later. In the scrotal sac, there is a cavity. This cavity is known as vaginal coelom. It is filled with a connective tissue which is called hydrocele. The cavity, that is vaginal coelom, is connected with the abdominal cavity. That small connection is known as inguinal canal. It is the same canal through which the testes have descended into the scrotal sac. The time when the testes descend is the 7th month of gestation period under the influence of testosterone. The copulatory organ or the external genitalia, that is penis, if its cross section is seen, there are 3 highly vascular tissues. The 2 posterior are known as corpora cavernosa and the anterior one is known as corpus spongiosum. It is corpus spongiosum through which passes the urinogenital duct. So, functions of all the structures and how these structures are present, what the role they perform, all these things we have talked of. So, this is the male reproductive system, the system part. Now, let us talk about the role of these glands, the seminal vesicle and prostate gland. They pour their secretions. So, seminal vesicle, seminal vesicles, they pour their secretion which contains fructose, citrate, some enzymes. These enzymes are coagulating enzymes. Coagulating enzymes. We will talk about these enzymes little later when we come to the stage of fertilization. After insemination, before fertilization, we will again talk about these enzymes. And there is one more substance which is present, is called prostaglandin. These prostaglandins, they help in the contractions of the female reproductive system, and these contractions will help the sperms move up to the egg. So, we will talk about this also when we come to the stage of that discussion of insemination and fertilization. So, it mainly contains fructose and citrates which are nourishment or nutritive parts for the sperms, enzyme which is a coagulating enzyme, and prostaglandins which are basically going to or are required for the contractions of the female reproductive tract. This is seminal vesicle. Then is prostate gland. The secretion of prostate glands mainly contain bicarbonate ions which maintain the pH of the complete secretion. Complete secretion means the secretion of seminal vesicle plus prostate gland and sperms, which is known as semen. So, we will talk about the semen in a minute, but they have bicarbonates. The secretion of prostate glands have bicarbonates. Enzymes. Here it was coagulating enzyme. In the secretion of prostate gland, it is anticoagulant or coagulating. It is anticoagulating enzyme plus citric acid, which is again nutritive tissue. So, seminal vesicles, their secretion, prostate gland and their secretion. There is one more gland, that is Cowper's glands. These Cowper's glands are also known as bulbourethral glands. They are also called bulbourethral glands. The reason urethral, because they are present at the junction of urethra and the vas deferens. Now, what is semen? Semen includes secretion of seminal vesicle plus secretion of prostate gland plus sperms. These three things, because the secretion of Cowper's glands are basically mucus, and that is meant for proper lubrication of the penis during copulation. So, that secretion is simply mucus. So, semen basically includes secretion of seminal vesicle, prostate gland, and so this is secretion of prostate gland and sperms. The pH of semen is about 7.5. That is, it is slightly basic. This is required, this pH is required to neutralize the acidity or acidic nature of urine, because sperms lose their functional capability in acidic pH. Urine is acidic. So, to bring it to neutral, this semen is slightly basic, and bicarbonates which are produced by prostate gland help in providing this basic pH. In one ejaculate, that means one time when these sperms are released or one time when the semen is released, the quantity of semen is about 3 to 4 milliliters, or let us write it here. One ejaculate, that means once when the semen is released, it has, or the volume is about 3 to 4 milliliters, and the number of sperms which are present in this 3 to 4 milliliters of sperms are 200 to 300 million. These many sperms are produced in one ejaculation, one ejaculate, which is about 3 to 4 milliliters. So, this completes the male reproductive system. We have just seen the system. We have not started with the first step, that is gametogenesis. Few more things are there which we need to discuss about the male reproductive system. Those we will take up at the time of either gametogenesis or fertilization. So, basic structure, which part performs what function, and what is the actual secretion of all these glands, and what is semen. So, this completes the male reproductive system. Now, next we will discuss the female reproductive system.